Today's video is about more complex examples of Punnett squares and genetics where we're not just dealing with complete dominance like we've looked at before. Complete dominance were those cases where if you have one copy of the dominant allele, then that's what you get. That will determine your phenotype. For example, if blue was dominant over yellow for a color of something, then having two copies of the dominant allele would mean that your phenotype is blue. Having two copies of the recessive allele would mean your phenotype is yellow. Or if you had one dominant allele and one recessive, you would still exhibit the phenotype of the dominant trait because that one wins out. But that's not always the case, and not every characteristic is quite so simple. Today what we're looking at is when neither trait is completely dominant. The first two cases are still pretty much the same. If you have homozygous dominant alleles, you have the dominant phenotype. If you have homozygous recessive alleles, you have the recessive phenotype. But the question is, what about heterozygous? There's a couple of different possibilities that the phenotype could exhibit for heterozygous alleles, and it just depends on what that trait is. So one possibility is that blue plus yellow could make green. They blend together so that neither one is totally obvious. The other option is that blue plus yellow could give me a mix where they're both still present, but I can pick out each one individually. So that might look like spots, or it might look like stripes, or it might just look like the two colors on two different parts. So here's an example of flowers. We have the dominant color being red, and we're going to use slightly different notation on the alleles just to show that these are all dominant. I can't really use uppercase and lowercase here. So I'm putting my alleles almost like exponents. My dominant red flower, my recessive white flower, and then my heterozygous options, where incomplete dominance shows a blend, so these made pink, and codominance shows a mixture, but I can still see both of the individual traits. In terms of Punnett squares, it's not any different than how we've already been doing them. So I put one parent across the top. So my pink had one allele for red and one allele for white. And a white flower had two recessive alleles for my white plants. Because I see that this is pink, I know that this is a trait where I get a blend. So this is incomplete. Then I do the Punnett square the same way. I look at what allele does it get from the top from one parent and what allele does it get from the side from the other. So these offspring of a pink plant crossed with a white plant, I have 50% have the heterozygous alleles, red, white. So 50% of these plants are going to be pink. And the other 50% are going to be white. I could do the same thing over here. This time I have 2 pink plants. Pink is heterozygous. So here I have 1 plant that is red. Here I have 1 that is pink. Here I have another one that's pink. And finally, I have another one, my 4th one, which is white. So I have 25% red, 25% white, and 50% pink. In this case, this is spotted. So this is codominance because I can see both of them. The Punnett square is exactly the same. So if you feel like you're getting this, you can jump ahead to the next section. If I have a spotted plant for one of my parents, I have 1 red allele and 1 white. And red for the second parent, red, red. And then I do my Punnett square the same way. And in this case, I have 50% of the offspring plants are red and 50% are spotted. I can make out both red and white on the plant leaves. One of the most common examples we use with codominant traits in humans is our blood types. And you may want to go look at page 34 in your textbook, the hardcover, to see where some of this information comes from. So type O blood in a human is recessive. Types A and B are codominant, which means for humans, there are 4 different blood types. I could be type A, I could be type B, I could be type AB, where you have both present, or I could be type O. So someone who's type O is recessive, and your book uses the letter I. So there's my lowercase I for the recessive trait. Their genotype must have 2 recessive alleles. Type A blood, I have 2 options because it is a dominant trait. I could either be homozygous dominant, or I could be heterozygous with the recessive trait. And then same thing for type B. I could be homozygous dominant, or I could be heterozygous with the recessive allele. And then finally, type AB blood is the codominant one. It's heterozygous because I need an A and a B, so there's only one way to do that. These Punnett squares work the same way as the ones we've been doing all along. So if I have one parent with type AB, they are going to give 1 allele that's an A and 1 allele that's a B. And then the second parent says they're heterozygous type A. So they have 1 dominant allele and 1 recessive. I know that's not a B because otherwise then they would have type AB blood. So finally, I do my Punnett square the same way. The allele from that column, the allele from that row. So these offspring here, we will have 50% that have type A blood. That's from this left-hand column. I will have 25% that have type AB blood from the top right. And then from the bottom right, the last 25% will have type B blood. And And I'll do one more example, but if you're feeling good, feel free to stop and go on to the next video. My final example here, I have one parent with type O. They must have both recessive alleles. And I have one parent with type AB. So by now we're very used to doing our Punnett squares of getting the allele from that row and column. And these offspring, 50% have type A blood, and 50% have type B. That's all for this video. I will see you guys in the next one.